If you want to learn how to design perfect primers for a standard PCR or real-time PCR, then this video is for you. In this video, I will show you the complete PCR primer designing workflow, practically and in a step-by-step manner, from getting your target sequence from NCBI to designing the primers using NCBI Primer Blast, and finally checking their specificity. We will also look at the key parameters you need to understand, including the primer length, the GC content, melting temperature, amplicon size, and primer specificity, all of which are important for designing effective primers. And by the end of this video, you would be able to design your own primers for PCR and also learn how to evaluate whether your primers are suitable for your experiment. So let's get started. For this demonstration, I will use a gene sequence that I have downloaded from NCBI. But you can also use your own target sequence as well. I am going to search for the HBB gene from Homo sapiens and select gene as the database type. It is important to be specific about the gene name, species, and the type of sequence you want to use. If needed, you can also refer to the published literature to confirm the correct gene name or the ID. I also recommend keeping the accession number of the sequence you use. This makes your work easier to reproduce and allows others to identify the exact sequence you used. Now let's get our sequence. If you are amplifying genomic DNA, choose the gene or the genomic sequence. But if you are studying gene expression using RT-PCR, you will generally work with transcript or the cDNA sequence. I will select and download both of them. The sequence will be downloaded as a zip file containing both sequences. Let's open it in Notepad++. Here we have the cDNA or the transcript sequence, and here is the gene or the genomic sequence. If I am designing primers for standard PCR using genomic DNA, I would use this gene sequence as the target. For real-time PCR, I would use transcript or the cDNA sequence. First, I will show you how to design standard PCR primers, and then we will design real-time PCR primers. The workflow is very similar with a few very important differences that I will explain along the way. For a standard PCR, I will copy this gene sequence into NCBI Primer Blast. Why Primer Blast? Because with Primer Blast, you can both design primers and check their specificity using Blast. So instead of designing primers first and then manually checking whether they match other sequences, we can do both steps in the same workflow. Here, you can set different parameters, but for most applications, the default setting works well. Let's focus on some important parameters. You can set the product or the amplicon size you expect to get after PCR amplification. For routine PCR, you can choose something like 100 to 1000 base pairs, depending on your application. For many standard PCR applications, the TM around 60 degree works well. The annealing temperature in the PCR cycle is usually set based on the primer's TM, which is often 1 to 2 degrees below it. It is also important to keep the TM of both the primers similar. Here, I am setting the maximum difference between the two primers to 1 degree. Now, to check primer specificity and make sure we do not get any unwanted PCR products, you need to select appropriate database and organism. For transcript specificity, choose RefSeq mRNA as database. This is useful when designing real-time PCR primers. Here, I will switch to reference genome. You can also change the organism from here. Since our target is human, I will check the primer against the human genome. Now let's open the additional parameter settings. Here, you can adjust the primer length. For a typical PCR, a primer length of around 20 to 23 nucleotides is a good range to minimize non-specific amplification and improve the specificity of binding. We also set the GC content between 40 to 60% for better primer stability. These settings helps to minimize template mispriming, self-complementarity, and complementarity between the two primers, which can lead to primer-dimer formation. We will leave other settings to their default. Next, click Get Primers. Now it is telling that the sequence is identical to the reference sequence, which is expected because we downloaded it from the NCBI. Let's submit it. This may take some time because the Primer Blast also checks the primary specificity across the genome. And here are the results. The specificity check shows that the primers are specific to our target and produce only one product. This graphical view shows where each primer pair is located on the target sequence. Below, you will find the detailed primer report including the primer sequence, TM, GC content, product size, and other parameters. You can also download the primer sequences in different formats. I will download them in tabular format. Now, although it is a text file, but you can easily copy the data to Excel. So this is how you can design the primers for a standard or the conventional PCR using a gene sequence. Now I will quickly show you how to design primers for real-time PCR using a transcript or cDNA sequence. I have copied the sequence into the NCBI template section, changed the product size to 70 to 200 base pair, selected RefSeq mRNA as database, adjusted the length, the GC content, and clicked Get Primers. And here are the target specific primers that you can use for gene expression studies using qPCR or real-time PCR. So this is how you can get your sequence, set the right parameters, and design specific primers for your target. Remember before ordering your primers that they are specific to your target, written in 5' to 3' direction, have similar TM values, and produce an amplicon size suitable for your experiment. And that's the complete basic workflow for designing PCR primers. And for more practical tips on research and publishing and AI tools, consider subscribing Explore Bio. If you have any questions, let me know in the comments and I will try to help. Thanks, and I will see you in the next video.